job. Okay, so a reaction that you can do on imines. It's called reductive amination, and it's an interesting reaction that we'll see again when we talk in our chapter on imines that's coming later on. Recall that you make an imine by taking a ketone or an aldehyde and reacting it with a primary amine under acid-catalyzed conditions. Probably you need to remove water because water is a product of the reaction. And the reaction gives you this interesting looking product, which is an imine. An I is for imine. It turns out we didn't do anything with imines, but they are electrophilic, and they are a lot like their aldehyde and ketone partners. That is, they can do addition and reduction reactions. Now, there's a lot we could get into. I only want to do one thing, and that is a reduction reaction on imines. You can use sodium borohydride, and I won't draw the mechanism for you. This is usually in a protic solvent. There are various conditions that are used, but in effect, what happens is sodium borohydride delivers a hydride equivalent to the carbonyl carbon. You could imagine drawing the BH bond below here and an arrow pointing to this carbonyl carbon, and then breaking the carbon-nitrogen pi bond with electrons going up on the nitrogen. That negatively charged nitrogen would then be protonated in a subsequent step. And your product would be this, which is now, instead of an imine, it is an amine or amine, or however you want to say it, an amino compound. Basically, this allows you to connect the carbonyl carbon of the aldehyde to the nitrogen of the amino group, and then the hydride from BH4 ends up here. And this is a convenient way of making amines. We'll talk about this more in a subsequent chapter. In some ways, this is related to a mechanism from chapter 13, or a reaction from chapter 13 that I told you to memorize. Perhaps you learned that you can take an amide and with lithium aluminum hydride and then aqueous workup afterwards, reduce the amide to the amine. Those two carbon-hydrogen bonds come from lithium aluminum hydride, and they proceed through intermediates that are similar to amines. We didn't learn that mechanism, but it's sort of a similar concept. Anything you want to ask about that? That overall series, make the imine in step 1, then reduce it to the amine, in step 2 is called reductive amination. Anything you want to know about that? I don't think you've got any practice problems on it yet. Yeah, go ahead. Is that a hand?
Does it have to be like that low H because water not working?
You'd want to use some solvent that can dissolve the imine and sodium borohydride. An alcohol solvent is an example of one of these. Not anything to memorize. Would water work? Maybe, if everything was soluble in it. Just will depend. Okay, others? Yeah?
Is there any difference between like the aldehyde and ketone and product?
No, for aldehydes or ketones, this would be the same. The only difference is if you follow through, if this were a ketone, this hydrogen would be another R group. Yeah?
I said we do the equivalent reaction with the ethylene as well.
Could you do this with enamines? Yes. You can reduce enamines, though we won't. Okay, others? Yeah?
Do you always do sodium borohydride?
Do you always do sodium borohydride? You could use lithium aluminum hydride. That would be overkill. Sodium borohydride's cheaper. Yeah. And there are other reagents that do this, but that'll be enough for now. Okay, back to chapter 16, a nucleophilic acyl substitution. The molecules we're going to talk about, we're sort of dealing with nucleophilic acyl substitution in biology. The molecules that we're dealing with are large enough and complicated enough that it's taking a lot of time for us to draw them. So what I'm trying to do today is spend not as much time drawing and more time talking about the chemistry. That will necessarily frustrate those of you that like to draw everything and get it down. And so feel free to abbreviate as you need to, and then understand the notes are going to be uploaded right after class, so you'll have access to them. Also, most of what I'm showing you is straight out of the text anyway, so that'll give you multiple places that you can go for information. We talked about acyl phosphates and thioesters last time. We just sort of barely got to thioesters, but I wanted to walk through it again. Making an acyl phosphate in biology often starts with a carboxylate nucleophile and then ATP. And as we pointed out, these bonds between phosphorus and oxygen are called phosphoanhydride bonds, and they're relatively weak. You can look at a phosphorus, any of these three phosphoruses, and think of them as basically reactive carboxylic acid derivatives. You can think of the phosphorus as being very much like the carbonyl carbon, and the oxygen as being very much like the carbonyl oxygen. So what happens in the mechanism that I showed you is that the carboxylate lone pairs will attack the gamma phosphorus to make, and then we will break the phosphorus oxygen pi bond. We will make not a tetrahedral intermediate anymore because now it's surrounded by 5 things, so I'm going to call that a pentavalent intermediate. But we will make that intermediate. Some of you have asked, how do you know which phosphorus gets attacked? The answer is you don't, but the enzyme controls that. And you'll learn more about that in biochemistry. But if the carboxylate oxygen attacks the gamma phosphorus, as shown here, you get this pentavalent intermediate, and then the electrons on the oxygen kick down and kick off everything else as a leaving group, such that your final product is the acyl phosphate and ADP. So here's still the ribose ring, the adenine base, and then the alpha and beta phosphates. All of that was a leaving group, which we're calling ADP. How can you tell it's a good leaving group? Well, you're used to thinking about phosphates as being conjugate acids, conjugate bases of some decently strong acids. The other thing that helps this as a leaving group is the fact that you've got the magnesium counterion, which I mentioned most of these enzymes use to help deal with the negatively charged phosphate groups. Then on the other side, you now have the gamma phosphorus attached to the carboxylate. So how is this like an acyl substitution reaction? In this reaction, the carboxylate is the nucleophile. I'm going to erase the orange there. Blue is what we use for the carbonyl carbon, and pink is what we use for the carbonyl oxygen. And then in green, we'll circle the leaving group. So nucleophile attacks, and then this is the leaving group, and that leaving group gets kicked off, okay? That gives you the acyl phosphate, which are intermediates all over the place in biology. Go ahead.
So the magnesium ion is really important for activating that gamma phosphorus, right? To
Yes.
let the reaction happen. But we also know that the reaction happens on the alpha phosphorus.
Right.
But as far as I can tell, like the alpha phosphorus and the beta phosphorus are pretty, like they look the same. So why is it selective to that alpha phosphorus and never the beta?
Right. So the question is, why does the nucleophile prefer to attack gamma versus alpha versus beta? And this is a complicated question. It's a good one. It's one that I probably don't have the answer to, but I'm going to give you just a couple of clues. First, you could imagine that the position of the magnesium ion, whether it's over here or over here, might control where the better leaving group is. Second, you could imagine that pentavalent intermediate where you got one phosphorus bonded to 5 things, 3 of which are negatively charged, is highly important, and that the enzyme likely has some groups around it that are stabilizing those negative charges, perhaps some electronegative NH bonds that it can hydrogen bond with a negatively charged oxygen. The enzyme can control which phosphorus is in which pocket that has which hydrogen bonding groups, and that can help activate one versus the other. That's not a very good answer, but it's probably all I can tell you at this point. You're not meant to predict what would happen because a different enzyme catalyzes attack of a carboxylate on the gamma phosphorus versus attack on the alpha phosphorus. So it's a totally different enzyme that does that. And it'll have a different name, and in biochemistry you would know what those enzymes are. Good, others? Yeah?
You might have talked about this already, but does this have to be in basic or acidic conditions, or can it be in any?
This is, the question is this in basic or acidic conditions? This is pH 7 or 7 point whatever is inside the cell is. 7.2, 7.4, I forget what the number is. Okay, others? Yeah?
Would the oxygen negative charge get protonated again after the reaction, or?
Would the negatively charged oxygen on either of these groups be protonated after the reaction? Is that the question? Yes, probably. I've shown them in the fully anionic states, but based on the pKa's of the phosphorus-containing acids we talked about, this one is probably about 50% dianion and monoanion. And depending on whether magnesium is over here, this one might get protonated as well. Generally, we just don't worry about it. We let biology do its thing, and we let chemistry take its course, but yeah. Others? Right, so that's an acyl phosphate. Now you've converted what used to be a nucleophile into something that itself has a good leaving group attached to it. This is basically biology's way of turning, remember we turned carboxylic acid into acid chlorides using SOCl2. This is biology's way of doing that. And now the carbonyl carbon now has a good leaving group on it, and you can now do subsequent nucleophilic acyl substitutions with this acyl phosphate. I want to just show you real briefly what happens when you attack the alpha phosphorus. So from our perspective and using color the way we've started, the carboxylate is again the nucleophile. This particular enzyme would activate the alpha phosphorus. So this is now going to be kind of like our carbonyl carbon. This is now going to be kind of like our carbonyl oxygen. And then all of this is going to be the good leaving group. And yes, you'd have magnesium over here between probably the gamma and the beta phosphoruses to help the leaving group leave. The mechanism is going to be the same. I haven't drawn it here, but electrons on the carbonyl oxygen would attack the alpha phosphorus, which we can draw in an arrow like this. And then that would lead to a pentavalent intermediate with a negatively charged oxygen on the phosphorus. Probably that intermediate is being stabilized by hydrogen bonding groups. Then electrons would kick down and kick off the green leaving group. Yeah?
What makes the left side a better leaving group than the right side?
What makes the left side a better leaving group than the right side? Good question. So I think the question is, let me circle in. I'm running out of colors here. I guess orange. So you're asking, why isn't that a good leaving group? Okay. If it left, this negatively charged oxygen would leave. It's low. That just I'll draw it real briefly like this. That's just an alkoxide. That's a non-resonant stabilized negatively charged oxygen. It is the conjugate base of an alcohol whose pK is around 16. So it's not a good leaving group. Whereas the pyrophosphate leaving group, these negatively charged oxygens are resonance stabilized, and the pKa's the pKa's of their corresponding conjugate acids are much lower than 16. It's a less stable leaving group because it's just a localized alcohol. Okay, what else? All right. So the product after this is what we will call an acyl AMP. Acyl is what we mean, I'll just highlight in gray. This is an acyl group, an R group with a carbonyl carbon, and that carbonyl carbon is now attached to something else. All of this is the AMP group. AMP stands for adenosyl monophosphate. And you can see it too is a good leaving group, much like the good leaving group for the acyl phosphate. But you can see that they're different, right? The acyl phosphate just has those two negatively charged oxygens. The AMP has one negatively charged oxygen and then this ribose ring with the adenine base. So the AMP is like a leaving group that has a handle on it that an enzyme could hold on to for specific purposes. The acyl phosphate is a much simpler leaving group. They have different uses in biology. Different enzymes will act on different acyl phosphates versus acyl AMPs. Okay, and I'll show you some uses for both of these in just a second. But before we do that, other questions? Okay, let's get down to the uses of these. So here is an example of an acyl phosphate. This is in amino acid synthesis. This amino acid on the left is called L-glutamine. It is one of the 20 canonical amino acids. Its one-letter code is Q, which is strange, but you run out of letters that make sense when you have 20 amino acids and only 26 letters in the alphabet. And nature starts with this gamma-glutamyl phosphate. Here's the amino acid part. This used to be a carboxylic acid, which nature modified with ATP to form the acyl phosphate. All right. So in pink, I've highlighted the carbonyl oxygen. In blue, I've highlighted the carbonyl carbon. I think I want to use green for the phosphate leaving group. Okay. That's the acyl phosphate. Your nucleophile is NH3. Now I'm going to put an asterisk on this because your body's not, there's no NH3 in your blood. If there is, you're very, very sick. So how is your body getting NH3 to the site of the reaction? It's using that PLP chemistry we talked about in chapter 14. It can deliver an NH3 using that kind of chemistry. You'll have to learn more about that in biochem because we don't have time to talk about it in a lot of detail. And I won't draw the mechanism here because you can think your way through it. Basically, I'll show you the first step. The nucleophile amine would attack the carbonyl carbon to get a tetrahedral intermediate. Now, right away, you're thinking, what about proton transfers, and can an amine attack a carbonyl carbon directly to give a negatively charged oxygen? Remember that when we've done stuff synthetically in the lab, we've had to pay attention to, do I need an acid catalyst or not? Understand that in biology, that's what the enzyme is for. So you probably have functional groups in the enzyme that are protonating or at least hydrogen bonding with the negatively charged oxygen as it forms. You probably have other groups that are removing a proton from the ammonia nitrogen, either as it's attacking or immediately after it's attacked. So this is why I'm not going to draw the mechanism, because I don't want to get into all of the details with the proton transfers. For now, it's enough to say the enzyme is handling that. But basically, you're going to get a tetrahedral intermediate eventually that looks something like this. And here's your phosphate group from your acyl phosphate. And then the negatively charged oxygen is going to kick down and the phosphate group is going to leave. And there we just made an amide bond in exactly the same way that we would do in the lab, only we're not using an acid chloride. And this is an example of the kind of easy chemistry I told you we weren't going to spend on any, that we do in the lab, that we're not going to spend any time on in class. We spent no time on acid chloride plus amine leads to amide, but that's exactly what we're doing here. The only difference is we've got a different leaving group, okay? So this is one way nature makes amide bonds in amino acid synthesis. Questions about that? Yeah?
On the intermediate, is it supposed to be an H2 or an H? Before or after?
At this stage, when I'm showing the phosphate leaving group getting kicked off, probably a base from the enzyme has removed the proton from NH3 that did the attacking. When we've done mechanisms for stuff that we would do in the lab, we've used an acid catalyst and its conjugate base to trade where the protons are to change who's the good leaving group, and that's probably also going on here. I'm just skipping ahead to a key step in the mechanism without showing all of that detail. Okay, others? All right. Yeah.
With the leaving group of phosphorus, is it now add on the AMP?
When the leaving group is phosphorus, what happens? What is the fate of phosphorus? one of the reasons you're breathing actually has to do with what happens to that phosphorus. That's aerobic respiration. You have your mitochondria. We'll learn about that metabolic pathway. We won't learn about how mitochondria take phosphate and ADP and synthesize ATP. But there is an enzyme called an ATP synthase, which will put that phosphate back onto ADP so we can go again with others. Yep. Okay. Miss anybody else? Okay. Here's an example of something that happens with not an acyl phosphate, but rather an acyl AMP. So I'm going to change my color scheme again. Here is the AMP portion, and then here is the acyl portion in gray. I've highlighted again the carbonyl carbon is blue and the carbonyl oxygen is pink. I'm calling this a fatty acyl AMP, not as an insult, but because the R group has many carbons in it. This is a 10-carbon long-chain carboxylic acid. It's called a fatty acid. It's part of metabolism. You can get it from triglycerides. Your body makes these. Your body also converts triglycerides into fatty acids. We've sort of talked about that a little bit. So a long-chain carboxylic acid that's in this case fully saturated is a fatty acid. So this is a fatty acyl AMP. As part of fatty acid metabolism or burning fat, your body needs to transfer this to another set of enzymatic machinery. And this allows us to talk about another carboxylic acid derivative that's very common in biology, and that is the thioester. So what biology does is, remember, what's in green is a leaving group. Biology has the thiol of this molecule. So how do you like that, right? You memorized it yet? That's basically ribose with adenine on it, a couple of phosphates. So you can see maybe where that part comes from, right? That looks kind of like ADP. Then a bunch of other stuff that we're not really sure about, and then a sulfur on the end. This is coenzyme A. This part is adenine, and this part is pantothenic acid or vitamin B5. So it's a nutrient in your diet. We call this coenzyme A, and mercifully, ever after, we will abbreviate it as CoA with an SH on it. Because the real business end of this molecule is this thiol, which is a nucleophile. Thiols are good nucleophiles. You learned that in 351. So what's going to happen now is exactly what we expect for a nucleophile and a carboxylic acid derivative with a good leaving group on it. We would expect the thiol to attack the carbonyl carbon. I'm not going to show that because you can push the arrow yourself or imagine it. Again, you might be thinking, can the neutral thiol attack that carbonyl carbon directly, or does it need to be deprotonated first? That's a great question. Probably the enzyme takes care of the proton transfer either before or during as the sulfur is attacking. Then, as I mentioned above, you would ultimately get to an intermediate that looked like this. I'm going to abbreviate that coenzyme A thiol like this, and then I'm going to abbreviate the AMP leaving group like that. Isn't that nice? Thank you for saying yeah. Yes. That makes it so much easier to draw. Here's our carbonyl carbon, here's our negatively charged oxygen, and then electrons on the negatively charged oxygen would kick down and kick off the AMP leaving group. What this has done is we have transferred the acyl group from AMP to the coenzyme A thiol. So I'll color in green the AMP group. And this is how we make a fatty acyl CoA. It's an example of a thioester. This functional group is a thioester. It's like an ester, only the oxygen's a sulfur. And this is an important intermediate in fatty acid metabolism. When you burn fat, your body can actually take this long chain carboxylic acid, which we're abbreviating here, and it's going to successively clip off 2 carbon units until it's gone. And we'll learn that chemistry in a subsequent chapter. But again, the core message is very simple. Nucleophile attacks, tetrahedral intermediate, leaving group leaves. And these are just biology's examples of leaving groups. Okay, questions so far? All right, and a mechanism is shown there in the notes, but so why would biology need thioesters? And the answer is they're a little bit less reactive than the acyl phosphates. So if you need, but they're also still decent leaving groups. So if you need something that can react like an acyl phosphate but hang around a lot longer than an acyl phosphate, thioester is a good choice. Biology uses both of them, and you've seen how biology can interchange them, interconverting an acyl phosphate into a thioester. Let me show you some uses of thioesters. Here's one that's relevant to neuroscience. So acetyl-CoA is the thioester where the acetyl group, which is just the 2 carbons with the carbonyl carbon, and then the coenzyme A thiol is the leaving group, reacts with choline, which is this 2 carbon amino alcohol where the amino group has 3 methyl groups on it and has a positive formal charge. And the enzyme will have the oxygen attack the carbonyl carbon of acetyl-CoA to get a tetrahedral intermediate. Electrons will kick down, kick out coenzyme A as a leaving group, and you end up making this new ester functional group, which is called acetylcholine, and this is a neurotransmitter. And then you also have other enzymes that are called acetylcholine esterases, whose job is to hydrolyze that ester bond between the acetyl group and choline. Another use of thioesters, that fatty acyl-CoA that we just made up here, in addition to being metabolized, can be appended to a lot of other molecules. So for example, when your body wants to ship cholesterol around, it doesn't just pump cholesterol out into the blood. Instead, it packages it in particles that are called HDL, lipoprotein particles, which you've heard of if you've ever seen results of a cholesterol test, right? HDL and LDL, does that sound familiar? What that is it's a large particle of lipids and then what I'm going to show you called cholesterol esters. So this is the large molecule known as cholesterol. If we're lucky, we'll get to how it's synthesized this semester. Please know. Notice that it's mostly nonpolar, but on this side it has an OH group. So it's polar on that side. And cholesterol fits nicely in membranes. However, if you modify this OH group as an ester with this acyl group, again, the coenzyme A is the leaving group, negatively charged, or the oxygen from cholesterol would probably be deprotonated by the enzyme, then it would attack the carbonyl carbon, you'd get a tetrahedral intermediate, and then electrons would kick back down and kick off coenzyme A as the leaving group. That would make this ester bond with cholesterol. That's called a cholesterol ester. And that's what's loaded into these HDL and LDL particles that carry cholesterol through your blood, from your liver to various tissues, back and forth. All right, all of those are what we will call acyl transfer reactions, which is just another way of saying nucleophilic acyl substitution. Okay, questions about that? Okay, so just to reassure you, because I know that at least some of you out there are like, oh my gosh, those were horrible molecules. I couldn't hardly even get myself to process what I was looking at. Do I need to have it all memorized? Clearly not. I've tried to highlight the important parts of the molecules in color and tried to illustrate what's going on. And inevitably, it's nucleophile attacks carbonyl carbon. You get a tetrahedral intermediate, and then the leaving group leaves. What I'm asking you to do is develop the skill of generalization. That means, can you look at something unfamiliar or new and see what it has in common with what you've seen before in other contexts. That's going to be essential to your success in biochemistry. Okay, other questions? I need a drink, so somebody better ask a question. Is there a particular reason why the test is only 2 days instead of more? Yes. 2 reasons. First, a lot of students will take it on the last day independent of whether it's 2 days or 3. So it's 2 so we can just all get on with our lives. I realize that's unsatisfying from your point of view. Second, for a class our size, the testing center will not let me open or close an exam on Saturday. That is because the testing center closes early on Saturday, and they don't want all of you showing up at 3 p.m. and saying, "Where's my test?" and then saying, "We have no chairs for you." So it's a couple of policy things. If that doesn't work out, you're welcome to email me. In general, people are generally able to find a window within a couple of days where they can take an exam. I understand that Saturday would be great, but they just will not let me do it. I could extend it through Monday, but then that's 4 days, and then we start, yeah, please, or like, please give me even more time. There is a point at which you need to face the music and take the exam, because I'm actually not going to slow down on Friday and Monday and stop teaching stuff. So it is what it is, and I'm sorry. But feel free to email me and argue your case. I'd be happy to hear. I promise not to delete that email. How's that? Okay. I want to talk now. Yes, go ahead.
Real quick, with that thioester,
This one? Not that one.
it is elsewhere, but what would you compare it to? Is it more like an ester or like a ketone? Oh, yeah, right here.
Yeah.
The alcohol group,
Good.
like a ketone.
Yeah, so esters, thioesters are more reactive than esters for sure. I would almost put them intermediate between like acid chlorides and esters, which sort of makes sense because Cl- is a better leaving group than a thiol is, a negatively charged sulfur is. I would put it basically at the same reactivity as sort of at the top of our list. We've got like acid chlorides, anhydrides, aldehydes, ketones, thioesters, and acyl phosphates can go in that list too. Yeah. So we'll see that reactions that exchange acyl phosphates for thioesters, we're not even talking about equilibrium constants and delta G's here, but just intuitively it should feel okay to trade an acyl phosphate for a thioester because they're about the same reactivity. So you're not really going uphill or downhill, it's just a trade. Whereas you might expect that going from a thioester to an ester would be downhill in energy and pretty favorable. Yeah?
Would you repeat what you said about why biology would want to use a thioester instead?
Yeah, thioesters are a little bit more, what we'll say, kinetically stable than acyl phosphates. They last longer. They're slower to react. And so you can hang on to them longer without... just think of it this way. The competing reaction with all of these activated carboxylic acid derivatives is the same competing reaction that you would have with any carboxylic acid derivative with a good leaving group, and that is water. The thing you want to worry about is water being a nucleophile, going in there, attacking the carbonyl carbon, and giving you a carboxylic acid as a result. That's the competing hydrolysis reaction. Water's pretty concentrated in our cells, and so we need things that are good leaving groups that are going to be slow to react with water, because if they react fast with water, we're wasting all kinds of ATP all the time to make these things. Yeah. Okay. Yeah.
My question is a little bit going back to the acetyl phosphate. So can it attack at the alpha and the gamma? Or how does
Can the carboxylate attack ATP at either alpha or gamma? The answer is yes. You're not meant to predict which. The enzyme's going to control which. So it's not fair for me to give you this starting material and this one and say predict the products, because how are you going to know whether you should make the acyl phosphate or the acyl AMP? Yeah. You'll learn more in biochemistry. The enzyme name will sometimes tell you what you should do. Yeah. Okay, now I want to talk just a little bit about amide bond formation in biology. So, and I want to do this in the context of what's called protein synthesis. You've also perhaps heard of it in other classes as translation. This is how your body takes individual amino acids and links them together to make amide bonds. So here is an amino acid. The side chain, we're not going to say what it is. You've got this positively charged ammonium group on one side. Here's a carboxylate group on the other. This is the alpha carbon, and this is the carbonyl carbon. Ultimately, if we wanted to convert this carboxylate into an amide, strategically we got to turn this carboxylate from a nucleophile into something that's got a good leaving group in place of that oxygen. And our strategy biologically is going to be to make an acyl phosphate derivative. So here we have again an amino acid and then ATP. The enzyme that does this is amino-acyl tRNA synthetase. You don't need to memorize that. You're just going to hear it again in your life, so I want you to have heard it here. It's an amazing thing. It's specific for the side chain. And that's actually why biology works. And that's some of the science and molecular biology behind how the genetic code works. But the amino-acyl tRNA synthetase recognizes this side chain, then it directs this carboxylate to attack the alpha phosphate group of ATP to get a tetrahedral, or now pentavalent intermediate with a negative charged oxygen. That negatively charged oxygen kicks back down and kicks off this as a leaving group. You've seen that before. And that generates this amino-acyl AMP. So that's not new. That's just a carboxylic acid, only now the thing on the other side, the R group on the other side of the carboxylic acid, is different. So this is an amino-acyl AMP. Then you might be thinking, okay, we're ready. Bring in that amino group of the next amino acid. We're good to go. No. The amino-acyl tRNA synthetase binds to a molecule called tRNA. Have you heard of tRNAs before? Ish. You know that at one end of the tRNA you have what's called an anticodon, which is 3 bases. And those 3 bases code for the amino acid that the tRNA carries, right? You ever wondered, how does the tRNA get the right amino acid on it? This is how. The amino-acyl tRNA synthetase recognizes the right tRNA and the right amino acid and brings them together. So first, again, you make the amino-acyl AMP, where now all of this is a good leaving group, right? Then the next step in the same enzyme is we're going to make an ester with one of the OH groups on the ribose end of the tRNA. Now I recognize that's terribly complicated, which is why I've highlighted the parts that are important. Carboxylic acid, carbonyl carbon, carbonyl oxygen, leaving group, nucleophile is that OH group. All of this is a bunch of RNA. There's an anticodon down here. This is the 3' end of the tRNA, if you know what that means, and if you don't, we'll talk about it later. Nature's going to have this 3' hydroxyl attack the carbonyl carbon to get a tetrahedral intermediate, which is then going to negatively charged oxygen is going to kick back down, and then the leaving group's going to leave. And that will give us now the amino acid attached to the 3' hydroxyl of the tRNA. And again, that is a physical connection between the right amino acid and then the tRNA that has the right anticodon. Notice that in this process, we converted something that was an acyl phosphate into something that's an ester. The ester is going to be more stable, so we expect this to be kind of downhill in energy. But this is also, it's really important that this connection be stable because we just went to all kinds of trouble to get the right amino acid and the right tRNA together. And again, if water were able to come in and hydrolyze this quickly, that would slow the process down and we'd be wasting all kinds of energy along the way. Okay, so I'm concerned. I'm looking into your eyes and I'm seeing desperation. Which is okay. A little bit of humility is good for the soul. But I know you want to know, what am I supposed to get from this? Go ahead. Why is it that the 3' OH acts as the nucleophile and not the 2' OH? That is a great question. The chemist is going to be like, I better use protecting groups. Biology is like, don't need them. And the reason is that the enzyme holds onto the molecule in such a way that it's the 3' OH that's close to the carbonyl carbon that it's going to attack. Yeah, others? Yes? Oh, not that far. Okay.
The molecule on the left, so it's two separate molecules that it's not its own OH group that's attacking itself.
These are two different molecules. This is a tRNA. This is an amino acyl AMP. Those are two molecules that are brought into the same active site of the enzyme. Okay. What are you supposed to get from this? What about that? That's the punchline, folks. That's the punchline. Have you got that? We're changing who the nucleophile is. We're changing what the leaving group is, but it's the same chemistry all the way through. That is the punchline. Yes?
Just a random question. Why does the amino acid attack the alpha?
Right. Why does the amino acid attack the alpha phosphorus instead of the gamma one? I don't know. My guess is, though, that the enzyme needs this portion here to hold on to the molecule. Okay. We still got a couple minutes. So let's ask, if you have more questions, let's talk. Yes?
Going back to the tRNA molecule, what makes it able to attack the oxygen on the carbonyl derivative instead of the positive charge?
Right. Why does, so the question is that the amino acyl AMP has a carbonyl carbon that's reactive, and then this group that should be somewhat acidic, and what controls that? The cop-out hand-wavy explanation in all of biochemistry is the enzyme does it. But what we really mean by that is these molecules fit into the pocket of the enzyme that's doing the chemistry that put the reactive groups in the right orientation to react with each other and not other things. Saw another hand over here. You're behind the computer. I can't hardly see you. Go ahead.
Going back and forth towards the beginning of class, when we were talking about lithium hydride and NADH4.
Hold on, folks. I can't hear. Say it again.
So when we were talking about lithium aluminum hydride and NADH4, are those interchangeable between those two reactions, or no?
Yeah, the question is on reductive amination, and could you have used lithium aluminum hydride here to reduce the amine? And the answer is yes. The bottom one, you need lithium aluminum hydride to react with the amide. Sodium borohydride is not good enough. Okay, I think that's all we have time for today. You want to come down?